大学校友会光华生命健康分会正式成立，是我们的校友积极践行中国特色社会主义大学的初心使命，对接健康中国的需求，服务国家战略发展的一次深入的实践，也是通过行业融合、产业融合，加快推进生命科学、基础医学、临床医学、药学、化学、大数据、信息科学、材料科学、管理科学、金融等学科的交叉融合，实现一个跨学科、跨领域、跨背景的资源整合与赋能的又一次重要的探索和实践。相信呢，我们生命健康分会的成立呢，一方面能够推动交流互鉴，交流非常重要，我甚至认为的话呢，是生产力的一个重要的组成部分。所以通过交流的话呢，能够进一步加强科技界、医疗界和产业界校友们的沟通、融合和合作。第二呢，是能够打造一个行业的生态的闭环，或者说行业的融合，能够打通临床医疗应用的需求端、创新基础学科的研发端、产业技术创新的驱动端，形成生命健康领域的行业闭环，展现生命健康产业的集群效应。另外呢，就是分会的成立呢，也能够进一步激发创新活力，推动生命健康领域科技的源头创新，为加快上海科创中心建设，为强化生命健康国家战略科技力量，为构建人类卫生健康共同体呢，做出积极的贡献。
这个复旦大学校名取自尚书大传，日月光华，旦复旦兮，预示大学是社会之光，与日月同辉。作为国家高等教育的学府，不仅应当研究学术、造就人才，同时呢，也要启佑乡邦，回报社会。
我们的分会呢，以光华为名，寓意分会能够践行行业使命感，弘扬社会责任感，面向世界科技前沿，面向经济主战场，面向国家重大需求，面向人民生命健康，为实现中华民族伟大复兴呢，贡献复旦大学的力量。那生命健康是一个永恒的主题，也是复旦人孜孜不倦的努力方向。今天呢，复旦大学校友会光华生命健康分会正式成立，是我们的校友积极践行中国特色社会主义大学的初心使命，对接健康中国的需求，服务国家战略发展的一次深入的实践，也是通过行业融合、产业融合，加快推进生命科学、基础医学、临床医学、药学、化学、大数据、信息科学、材料科学、管理科学、金融等学科的交叉融合，实现一个跨学科、跨领域、跨背景的资源整合与赋能的又一次重要的探索和实践。相信呢，我们生命健康分会的成立呢，一方面能够推动交流互鉴，交流非常重要，我甚至认为的话呢，是生产力的一个重要的组成部分。所以通过交流的话呢，能够进一步加强科技界、医疗界和产业界校友们的沟通、融合和合作。第二呢，是能够打造一个行业的生态的闭环，或者说行业的融合，能够打通临床医疗应用的需求端、创新基础学科的研发端、产业技术创新的驱动端，形成生命健康领域的行业闭环，展现生命健康产业的集群效应。另外呢，就是分会的成立呢，也能够进一步激发创新活力，推动生命健康领域科技的源头创新，为加快上海科创中心建设，为强化生命健康国家战略科技力量，为构建人类卫生健康共同体呢，做出积极的贡献。
这个复旦大学校名取自尚书大传，日月光华，旦复旦兮，预示大学是社会之光，与日月同辉。作为国家高等教育的学府。
So, hello everybody. Good morning and good evening to Dr. Wei. So we have a 12-hour difference. And today is my great pleasure to host Dr. Wei's lecture. This lecture, the organizer is Dr. Tan Weihong. I'm the co-organizer for this lecture series. I joined with lecture host Zhao Yongchao, Li Gaopeng, and Cao Qin, right? Cao Chen, right? Zhao Chen, let me see. sorry, that's Wu Chen. So I'll give a very brief introduction about Dr. Wei. Dr. Wei is a world-renowned scientist in the cancer research. And he graduated from Shandong University in 1993 and received his master's degree from a Chinese Academy of Science in Beijing and received his PhD degree from the Brown University in 2002. And after that, he joined the Dana-Farber Cancer Institute as a postdoc. And his mentor was Bill Killing. As you know, Bill Killing is a Nobel Prize laureate and won the Nobel Prize because of his contribution in oxygen sensing and also the HIF-1 alpha regulations. So in Chinese, 我们的魏教授是师出名门啊。So Dr. Wei is currently holding a professor position at the Dana-Farber Cancer Institute and Beth Israel hospitals. His primary interest is post-translational modification in cancer development and progressions. He received a number of honors and awards, and including in 2022, he was elected as a member of Academy Europaea. In 2015, was elected as a fellow of American Association for the Advancement of Science. In 2020, he received a very competitive NCI grant that's called NCI Outstanding Investigator R35 Award. So only very few investigators can receive this big grant like R35. And today he's going to talk about his recent discovery in protein post-translational modification in cancer therapy. So with that, please join me to welcome Dr. Wei.
Okay.
Wei. Yeah,
Thank
okay.
you. I try to share screen. Okay, let me see. I want to share screen. I don't know why I cannot.
Well,
I'm sorry.
I try to find that link.
I choose to share screen, entire screen window. I'm sorry. I'm sorry for this. I should choose to what to share entire screen. Okay, share.
Control up at the bottom you can see participant chat and share screen. There's a green on my computer. There's a share screen in green color. Did you see it at the bottom?
I have the share screen, but I don't know why I cannot.
Oh, do you need to log out and log in again?
Yeah. Why did you log off?
You can log out and log in again. Give a try.
Okay, let me do it again. Oh.
我们申老师来了。
我们老师是这个大同行，又是小同行里面的专家。今天你们是，你一定要 open the your audio。
申老师，你没有开话筒。
OK， 好，那现在开了。
难怪我们没听到你讲了这么多的好话。
对，讲的都，我是说你那个 studio 那个打网球还是打高尔夫的，是 pretty good 的，在哪里搞来的？啊，我们里面你看看穿的这么厚，我们的 T 恤穿上了，是不是？这个年轻人跟年纪大的就是不一样。
说了一批这个适当的。
我们把文艺给 kick 大过了 ，speaker 都不能讲话了，你看看
这个 share screen 有点 problem。
是啊，这个网上很多人啊，我们不能乱说话哦。对。
你可以给我们讲讲这个 ubiquination 的历史，是吧？
对。
还有得了诺贝尔奖。
对啊，这个诺贝尔奖2004年得了嘛
？04年04年。
这个 chemistry 01年是得的 Alaska。04年得的诺贝尔奖，为什么04年得了诺贝尔奖？就是因为03年那个苯地美唑被 FDA approve for 那个 treatment of multiple myeloma。proteasome inhibitor。所以它一般的话就是 breakthrough for the basic research. If advance to drug discovery and really benefit the human being, and you have a very good chance to get Nobel Prize. All right.
这个现在的 PROTAC 也是针对 ubiquination 吧，然后通过 ubiquination 这个 tool 来 target the subject。
Yeah.
现在是有几个advance 进入了第3期。
对，已经好几个进入第3期了。有好几个进入第3期了。文业。
是这样，我 zoom in 那个用那个 meeting number 和那个 password， 它就不让我进，我只能log on as a web format， 就是通过那个那个 browser format。browser format
我不知道你能够share screen， if you can share screen and that will be fine。
我是从 WeChat log 进去的，看来样子是
就是这个the meeting 和那个 password number 为什么不可以 log in 呢 ？International 不可以吗？
对，我不晓得，我 log 不进，他说是 incorrect number， 所以我进不去。
你是 type in 吗 ？type in 的话是不是能好一点？不要 copy paste。
我再试一下。
因为刚开始我也是没有 log in， 但是我 type in 一会就进去了。
是吗？
你 type in。
这个 share screen 是在我看是在 bottom。
你不能放那个 noise。
share screen 的时候，我们孙老师可以继续讲给我们。
那就好了。你不要一天到晚让我多说。
Sorry. Technical problem. Yeah, good. Now.
So yeah, now we get it.
Very good.
Great. I'm sorry. Yeah, I was not able to log in at the beginning. Yeah. Okay. Good. Figure it out. Sorry for the delay, and so it's typical, like doing science. Yeah, always having problem. Okay. Thank you, Jermaine and Wei Hong for inviting me to this seminar. And today, I talk about my lab work on targeting protein post-translational modification pathway for cancer therapy. So basically, we know that the way we do in the precision medicine era, we want to distinguish normal somatic cells versus cancer cells and kill the cancer cells specifically. And to this end, actually, Dr. Hanahan and Bob Weinberg summarized a dozen of cancer hallmarks that serve as a guide for us to kill the tumor cells. And in my opinion, there are two features about these cancer hallmarks. Number one, they're supposed to be driven events. Number two, they can be targeted, or in other words, they are pretty much enzymes that can be targetable. So most of the prevailing cancer targets are actually happen to be, most of them are components of the known cell signaling pathways, or PTM components, WISO. We all know that our human body contains 25,000 genes. That's all we have, not that many. And then through the transcriptome expansion, we can have up to 100,000 transcripts. And then the magic happens upon different kind of stimuli. Every nascent transcribed protein receive PTM modification, and that can push the total proteomes to over 1 million. So this diversity is critical because that allows our body to be able to respond to upstream experimental cue or environmental cue. And so we can respond specifically in order to certain stress or certain stimuli. And more importantly, if this pathway was mutated, some disease might happen. In here, I give a short list of all the known or well-characterized post-translational modifications, but the list is ongoing right now. And for all the PTMs, we know, and my lab personally, I know Jimin also work on ubiquitin, methylation, acetylation, and protein hydroxylation, punctuation, and phosphorylation. And for all those PTM, they share features. They do have a writer that can add the PTM on the substrate protein, and they have an eraser that can remove it, and they typically have a reader that can read it. And most importantly, those typically occur on protein, and those modification has downstream biological functions. And you can imagine all the mutation along those pathways potentially can cause human disease, including cancer. And as I mentioned, a lot of those PTMs occurs on the known tumorigenesis-related proteins. For example, here I borrow a slide from Dr. Alex Tucker, who is my next door neighbor, who summarized all the known PTM on AKT protein. And my lab has contributed by identifying that AKT can subjecting to CDK modification during cell cycle, and allow the cells to have elevated AKT in S phase. Also, we found out that actually this collaboration with my mentor, Dr. Bill Kalin, that hypoxia can lead to activation of AKT by reducing AKT protein hydroxylation, which is another PTM that can suppress AKT function. That explains why in the center of the tumor, which typically experiences hypoxia, that's elevated AKT to push for tumorigenesis. And we also identify as lysine methylation dependent activation of AKT. And you can imagine all those aberrant PTM push for AKT overactivation that favor tumorigenesis. And on the flip point, PTM also occurs on tumor suppressor protein. For this, I borrowed a slide from Dr. Pierpaolo Pandolfi, a previous Cancer Center Director of BIDMC. And you can see here, again, P10 also can subjecting to many layers of PTM regulation. My lab has contributed by showing that P10 can undergo lysine methylation or undergo WWP1-mediated K27, a typical ubiquitination. This ubiquitination does not lead to P10 degradation, but rather P10 inactivation. And therefore, in most tumors, over estimated 40% of the carcinoma, P10 activity is reduced, or P10 expression was reduced, that contributing to tumorigenesis. So you can see here that the prevailing hypothesis I have is that for all the tumor cells that's aberrant in cell signaling pathway that leads to aberrant protein translational modification, either leading to protein homeostasis or protein function defection, and that leads to cancer. And if we can restore those PTM, we pretty much can potentially targeted kill tumor cells, but not normal cells. So that's actually pretty much idea during the last 2 decades for human beings that to actually develop almost all different kind of inhibitor against all cancer hallmarks. And I want to emphasize that potentially there's also crosstalk between cancer hallmarks. I'm going to talk about the crosstalk in a recent paper we published in the later stage of the seminar. So as I mentioned, the major lab research SAM since I'm a PhD and then a postdoc is to targeting the aberrant cell cycle signaling pathway for cancer therapy. And this is pretty much the pipeline of all the CDKs in our human body. Every single CDK we have an inhibitor behind it, and the CDK4/6 dual inhibitor has been FDA approved for treating human cancers, including breast cancer. But if you really take a closer look of the cell cycle, we typically call cell cycle engine or cell cycle machinery. It operates like a machine, unidirection. And this cell cycle machinery was fueled by this periodical cycling CDK activity. And this actually, in part, was driven by periodical protein destruction of the cyclins. That's where they got the name, because their expression expresses periodical during cell cycle. And exactly anaphase promoting complex that targeting the mitotic cycling, like cyclin A and B for destruction in M phase, and the SCF complex targeting the G1 cyclins like cyclin D and E for destruction in G1 to S transition. So you can see the better we understand the degradation machine, the better we can understand the whole cell cycle become aberrant in human cancer, and better we can design a drug to kill the tumor cells. So what is ubiquitination? Ubiquitin is a 76 amino acids tag through this E1, E2, E3 enzymatic cascade can add it onto the substrates, and depending on the topology of where the second UB was added, you can divide into different linkage, and K48, K11 linkage protein destruction signal, but K63 or linear lineage is not destruction, but rather enzymatic activation signal. So for the last 2 decades, the whole field thinking about how to targeting the UPI system for cancer therapy. And the first idea jump into minds, everybody do that, is to inhibiting the system. How to do so? We can targeting the 2E1, the 38E2. Somebody said 600, and somebody said 800 E3 ligases. And also we have over 100 DAPs. That's pretty much the portfolio of the enzymatic pathway on this field. And so what we think is that if you really want to targeting the system, you better understand the physiological contribution and the detailed mechanism. And that's why when I was a postdoc in Bill's lab in Dana-Farber, as Jimin said, I mean, the first project I really study is to understand the oncogenic role for SCIP2. And we found that SCIP2 expression level actually fluctuates during cell cycle, but if you depleting the upstream E3 ligase, in this case, CDH1, SCIP2 become stabilized. Now we know that makes sense because if you look at SCIP2's downstream target P27, expression is inversely correlates with SCIP2. But if you have high SCIP2, then P27 level become very low and cells enter S phase ahead of schedule. So therefore, we were able to show that APC CDH1 suppress SCIP2, and that actually implicates CDH1 potentially can function as a tumor suppressor. And later work from my own lab show that very like the histone code, there's a SCIP2 code as well. So in the N terminals of SCIP2 can subjecting to different modification, including acetylation, phosphorylation, and those modifications can allow SCIP2 to escape CDH1-dependent destruction process, and that stabilized SCIP2 can favor tumorigenesis. So, and also we studied tumor suppressor function of FBW7. So that was roughly 20 years ago. And the work from Bert Augustine's group in Johns Hopkins in Science paper showed that in colon cancer setting, FBW7 was the number 4 most mutated gene in colon cancer. And when Ron DePeno was then at Dana-Farber, that was 2007 Nature paper, they show that in one specific cancer types called T-cell acute lymphoblastic leukemia, the FBW7 mutation rate is as high as 30 to 40%. And the mutation clustered in the WD40 repeat domain, that hotspot mutation typically involved in disruption, the protein-protein interaction of the substrates. So when I was in Bill's lab, I was able to identify Sejong as a FBW7 substrate, and other groups defined Notch1 and CMYC as a substrate. Sejong, Notch1, CMYC, we call it early immediate gene. So those genes will be elevated upon growth factor induction. And on one side, they induce cell growth. On the other side, they can induce cell death. And you can see here, when you lost FBW7 and MCL1 go high, counterbalance this process. Therefore, they live on the edge. And if you have an MCL1 inhibitor, you will kill only the FBW7 null cells, but you don't touch the wild type cells. We call it synthetic lethality. And this is a perfect example of how the benchwork study can translate to bedside. So our human being has 68 adbox protein. So we think eventually we'll have 3 baskets. So if it is an oncoprotein like SCIP2, we should develop inhibitor like Huikuan and MD Anderson did. If it is at BW7, I don't think we can do any gene therapy right now. We can do is to using synthetic lethality and find the most important inhibitor, downstream oncoprotein, in this case MCL1, develop MCL1 inhibitor to kill the BW7 null cells. Or if this context dependent like beta TRCP, the patients need to be aware, you might be able to retard the primary site tumor, but you might ignite second site tumor because the function in tissue are really context dependent. All right. So as I said, the first idea for treating tumor is inhibitor, but second, we know in Chinese we say 大禹治水. So the idea is that instead of inhibiting, we might be better to harness this system. So how to do that? This idea was created by Dr. Craig Prose and Rudy Shea in 2001. You involve the generation of a functional chemical chimera. One end is an E3 ligase ligand that can bind to E3 ligase. Then there's a linker links to a warhead that binds to any protein of interest you want. AKT inhibitor, P1 inhibitor, anything, CDK4 inhibitor. And this can allow the E3 to be dragged into the protein of interest and triggers ubiquitination and subsequent degradation. So we all know central dogma. Our gene was encoded on DNA, translated into mRNA, then translated into protein. When we have a human disease, that typically, like cancer, is occurring on the DNA level. So there are 3 ways we can do. We can using CRISPR to correct the mutation on the DNA level. It's irreversible. So number 2, we can use RNAi to convert at RNA level. Or number 3, you can use PROTAC to correct on the protein level. And in my mind, this PROTAC is more like a drug, so has more potential to be translating into a powerful, potent anti-cancer therapy. So one thing I want to really point it out is unlike the reversible inhibitor, which typically the frontline treatment, like EGFR inhibitor, AKT inhibitor, that's what we use. That's what we know typically for treating cancer. PROTAC is catalytic degrader, therefore it has catalytic ability. So it's like, So the burning question is how to timely and spatially control the degradation power of PROTAC. It turned out that we know how to do that. In our routine laboratory work, when we're generating genetic knockout by knockout, we know that if you knock out an essential gene like KRAS or PI3K, it's typically embryonic lethal. The mice cannot survive. How to do that? We use Cre-Lox system. We can cross with a Cree that express only in specific tissue, so we can creating the tissue-specific knockout. That can allow us to study lung cancer, breast cancer, pancreatic cancer specifically. So the question will be, can we develop a conditional PROTAC that degrading the bad guy, the oncoprotein, only in the tumor cells, but not in the normal cells? That will be really ideal. How to do that? We were inspired by the new technology called optogenetics, that you can use UV to activate a neuron in a very specific manner. So in collaboration with Dr. Jianjin's group in Mount Sinai, we developed this optoprotect. We added a cage on the pomalidomide, the E3 ligase ligand, that made it inactive. Upon UVA treatment, that's undergo optolysis, and then we were activating this only in the cells we wanted, and then causing the protein destruction. So this is the co-crystal structure of pomalidomide with cereblon E3, and that's where the critical bond was forming, and that's labeled here red. You can see here we can edit a caged compound that can pretty much interfere with the interaction. So this was created by Dr. Jianjin's group at Mount Sinai, and we are using a protocol that has been optimized in this way. In the darkness, the pomalidomide is pretty much inert, not functional. But upon UVA treatment illumination, the inactive form will be activated because they undergo this called optolysis and then decaging. So the idea is we are using this protocol developed by Dr. Bill Killen in their 2014 Science paper, where they characterize pomalidomide as a molecular glue that can induce the E3 cereblon and the substrate RKZF1 interaction. You can see here, pomalidomide indeed can do the job, but if you use optopomalidomide, the binding is gone because we know we occupied the interacting module. And then if you do a UVA treatment for a short time, the binding is restored. And if you look at this destruction, pomalidomide can degrade in RKZF1 and 3 in a dose-dependent manner and a cereblon-dependent manner. But if you use optopomalidomide, there's no destruction, even in over 100-fold concentration. And if you're using UVA illumination in vitro for as short as 5 minutes, actually 15 minutes, 30 minutes can all do the job, the degradation is restored. We did use another method. We converted the DBAT created by the DBAT, which is a prototype of the most of the PROTAC. We also did that. Jay Bredner creating DBAT. We added this cage. You can see here DBAT can degrade in BRD3 and 4 in vivo in cells, and opto DBAT cannot do that unless you do UVA treatment for 15 minutes. So we created a platform, we call it optoprotect. We are confident that all the pomalidomide-derived PROTAC can behave very similarly. So in which way they are inactive. Upon UVA treatment, you can activate the PROTAC to do the job you want them to do. In this way, we added a switch or dimension. We can, using UVA, to give the order at the tissue we want, at the time we want for the destruction to occur. But one thing I want to really point it out immediately, UV has limited tissue transmission ability. So if you have a deeper tissue disease like breast cancer, you may not be able to expose to that. So how to overcome that? One way to do that is folate PROTAC. We actually use this idea because of cancer metabolism, inspired by Jimin's work. We know the tumor cells love folate. And so they do with that, they will express folate receptor on the surface, so they can uptake a lot of folate to feed their glycolysis process. Yeah, as a matter of fact, tumor cells has a folate receptor expressed, and normal cells don't have that. In this way, we're using folate as a guiding missile to guide the folate attached PROTAC into the tumor cells enriched, and the tumor cell will enrich for this and go cleavage, and then we are activating the PROTAC to degrade the tumor cells, but normal cells doesn't do so. Another way, actually, we were inspired by my visit of Yi Sun's department in 2019, and I think the chair asked why we don't use gamma irradiation. Such an inspiring statement. It took us 4 years. I can tell Yi, we just figured how to do that right now. So now we are working on gamma irradiation-induced PROTAC activation. That's another way to overcome this. So another thing I want to really point it out, for majority of the PROTAC, if you people tell you they do a PROTAC, turn existing inhibitor to a degrader. You have to have an existing inhibitor. If you have no inhibitor, no chemical ligand, there's no way to creating any PROTAC. How to overcome that? I actually figured out one way to do that is by taking advantage of the protein's biological function. So in this case, we created a TF PROTAC for targeted destruction of any oncogenic transcription factor to expand the protectability. How to do so? Well, we know that majority of the transcription factor, their main goal is to bind to a DNA motif and then trigger gene transcription of the gene that contains the motif. And we also know that a lot of oncoprotein are transcription factor like c-MYC, HIF, beta-catenin, YAP, the list ongoing. But most cases, transcription factor is very hard to develop inhibitor. They don't have a drug pocket. So what we did is that we take advantage of the in vitro click chemistry that created by Bertuzzi Labs, and you can see here we're using in vitro chemical click reaction to link E3 ligand VHL to a DNA motif. So therefore, we can recruit the E3 to transcription factor to degrading the transcription factor. So we take FKB as a first example. We're using the validated decoy motif binding to FKB, and then we actually basically adding the linker and link to the VHL. We screen for the linker. Yeah, you can see here number 15, number 16 are very potent in degrading endogenous P65. So we are able to creating, in theory, if you have a transcription factor with a known DNA motif, you pretty much can develop a degrader for that. And the number is our body contains over 1,600 transcription factor. Among them, over 1,200 has experimental defined DNA motif. So therefore, there's a lot of information on that that potentially can guide us to develop those TF PROTAC. So we all know that become tumor is not a one single step. Work from Dr. Bert Augustine's group show that it takes multiple genetic alterations that typically involve gain of oncoprotein expression, reduce or loss of tumor suppressor. So we know that if you develop PROTAC, allow you to targeted degradation of the oncoprotein, the bad guy. But then another part is that can we develop the DAPTEC for targeted stabilization of tumor suppressor? And we all know the big three tumor suppressor, PP3, P10, RB, and the list ongoing. So can we really take advantage of this to potentially also develop more novel targets for cancer? So we think that the PROTAC and the DAPTEC will complete the toolbox to manipulating protein abundance for targeted cancer therapy. How to do that? I'll give an example, a very simple example. Here we're using the same trick we developed. We find a chemical ligand binds to DAB. Here is O2B1 linker that was identified by Dr. Dan Numura group in UC Berkeley. And then we use in vitro chemical click reaction to link it to a DNA motif. And this time the DNA motif belong to a tumor suppressive one. In this case, it's either PV3 or FOXO. So you can see here, this is a DNA binding motif for FOXO1 and 3. They share a quite similar DNA binding motif. And we're able to show that the DAPTEC are able to stabilize FOXO3. To a less extent, FOXO1, the reason behind that, we don't quite understand. There's some specificity there. So therefore, we think if you really, really want to target in the UPS system for cancer therapy, you really need to look at both sides of the coin. You can either inhibiting the UPS or hijacking DAP, DAPTEC, that serves to stabilize tumor suppressors. Or you can harnessing or hijacking the system through PROTAC or using DAP inhibitor, and the goal is to really achieving targeted destruction of oncoprotein. Or you can combine these two approaches together that give you the power for precision medicine. So besides focusing on cancer intrinsic pathway, so I think nowadays we know that we appreciate more that the tumor become tumor not only because of the intrinsic pathway, but also become the tumor microenvironment. And more than that is to escaping the immune surveillance that allow our body not be able to attacking the tumor cell that typically contains a mutated genome. So how to do that? So I will give you an example of how to combine CD4/6 inhibitor with PD-1 blockade for cancer therapy. So this I borrowed a review article from my longtime collaborator, Dr. Gordon Freeman. So they summarize all the ligand and the receptor that involved in modulating immune response. And I really make it simplified. I only focusing on the PD-L1, PD-L2 as a ligand binding to PD-1 as a receptor. One reason behind it is because the PD-1 antibody or PD-L1 antibody has been shown to be clinically very strong in inhibiting cancer or boost the cancer immunity. However, even that, we know that the response rate are not promising, except in certain cancer like Hodgkin's lymphoma. Majority of the cancer, actually, the response rate is only like around 25%. And that's a paradox. The tumor cells express higher level of PD-L1, typically responding better to PD-1 or PD-L1 blockade antibody treatment. So people has been really struggling or try to striving to augment the immunotherapy response, try to turn cold tumor into a hot tumor. So how to do so? There are many ways to do that. You can combine immunotherapy with radiotherapy, chemotherapy, targeted therapy, all kinds of treatment. But the point is that how can we have the prediction power and the prognostic power, and how we really can predict whether this patient should use this one strategy and the other patient should use the other one. My argument will be the more mechanism we know, the better we can have the knowledge to guide the treatment. So if you look at PD-L1 regulation, there's many layers. There's transcriptional regulation, there's translational regulation. Yeah, the key here I want to, as a person studying cell cycle, I want to know is the physiological condition, such as cell cycle, whether can affect PD-L1 level. And strikingly, we found out that indeed PD-L1 level fluctuates during cell cycle. And we know, as I mentioned in early presentation, the cell cycle was driven by CDK, many different CDK. So the key here is which CDK might play a role in do that. And to answer that question, we actually collaborate with Dr. Peter Sensinski in Dana-Farber, whose lab extensively studied all the CDKs. And long story short, we found out the answer. Cyclin D1, D2, D3 triple knockout maps has hugely elevated PD-L1 level. But if you look at cyclin A1, A2 knockout, no. Cyclin E1, E2 knockout, no change. We know cyclin A, cyclin E binding to CDK2, cyclin D, 1, D2 binding to CDK4, cyclin D3 binding to CDK6, and strikingly, CDK4 knockouts MAFs elevating PD-L1, CDK6 knockout does not. So there indicates that it's CDK4, cyclin D1 or D2 might be the kinase doing this whole thing. So luckily, CDK4/6 dual inhibitor FDA approved for treat cancer. We're using palbociclib that's developed by Pfizer, and we're treating RB positive cells. That's heavy elevation of PD-L1, like the CDK4 knockout MAFs. And if you're using this treating RB deficient cell line, that's also elevation. And more than that, CT2641 are mice-derived tumor cells. Palbociclib, ribociclib, and amibociclib, all known CDK4 inhibitor elevating PD-L1. So that's very very interesting, because we know that PD-L1 is an oncoprotein. Elevated PD-L1, you will expecting to help the tumor cells evade immune surveillance. So basically, we actually using a mice as a potential vesicle. We actually treating both control of palbociclib for 10 days, and then harvest the mice. We harvest all 16 different tissues, and we found out that in every single tissue we harvested, palbociclib treatment can elevating PD-L1. So we concluded that palbociclib can lead to systematic elevation of PD-L1 in all tissue that potentially be. You can imagine if a clinical trial of human being, a breast cancer patient, receive palbociclib, potentially similar thing will happen. That's a bad thing, but meantime, as I mentioned to you, elevated PD-L1 potentially leads to enhanced response rate to PD-1 blockade. Is that a potential outcome? I'm not going to talk about the mechanism here. It involves a lot of work, but I'm talking about the preclinical study we actually did together with Gordon's lab. We're using 4 conditions and treated, so either control or the PD-1 antibody alone, or the CDK4/6 inhibitor alone, or combination therapy. And then we look at how this affect the survival and tumor size. You can see here, for the tumor, if you doesn't do anything, all 13 mice die within 20 days. If you're using PD-1 antibody treatment, roughly 20 to 25% of the mice survived. If you're using CDK4/6 inhibitor treatment, there's a little bit prolonged the mice survival, but eventually they all die. But if you're using combination therapy, 75% of mice survive. That's the surviving curve. So in addition to this, we also explored all kinds of PD-L1 modification, and we found out that like many other proteins, PD-L1 is subjecting to various different modification. As a matter of fact, if you're using HDAC inhibitor, you can block PD-L1 nuclear translocation as a way to actually suppress tumor regenesis. You can also control PD-L1 destruction by either AMPK agonists, V1 inhibitor, CDK4 inhibitor, as I described, pumptolation inhibitor, METDL13 inhibitor, or PARP inhibitor. In those cases, you can impact PD-L1 level as a way to influence the cellular response to immunotherapy. Or you can block PD-L1 secretion out by using NADD4L inhibitor. So all those can allow us to target achieving PD-1 level changes as a way to interfere with immunotherapy. So as hopefully I have convinced you that indeed by interfering with all kinds of PDR modification, we are able to potentially retard the tumor cells by affecting its biology and then restore its ability, kill the tumor cells. That involves in developing all the inhibitors or many other ways. And I don't have enough time to talk about our work involved in methylation or acetylation, but believe me, all those important modifications typically associated with aberrant function of the key enzyme that leads to tumorigenesis. And that's the reason I actually proposed an idea, we call it engineered PDM. So the idea, you can see here, you drive a car, you buy a new car, brand new, everything is great. But when you drive for a while, it falls apart. And then you take your car to a dealer and repair only the place that's wrong. The same idea, every single tumor cells, after a long time, their PTM was wrong. And the idea is that if we can identify what PTM goes wrong, and then we're using the bifunctional chemical. I give you an example of the PROTAC and DAPTEC, but think about every single pair of the PTM. There's a yin and yang regulation. You can hijacking the enzymes and the writer or the eraser. You can achieving a targeted repairment of the PTM, and that gives the minimum toxicity issue to the normal cells as a way of precision medicine to kill the tumor cells specifically to allow us to do that. With the power of DNA sequencing, we know more and more, a deeper knowledge of what's wrong with the tumor cells. We'll better develop a more targeted way to restore the signaling pathway and then kill the tumor cells without touching the normal cells in a substantial way. So I will really acknowledge all the people involved with the work. I think most of the PROTAC work was done by Dr. Jing Liu in my lab. All the early work on epoch protein was done by Damin Gao and Hiroyuki Inuzuka in the lab. All the immunotherapy work was done by Yinfang Zhang and Xiaoming Dai in my group. And we have many collaborators in Boston, Houston, and China. Dr. Yi Sun, Dr. Jianjing, Dr. Zhiming, and Michele, Alex. So we really appreciate everybody's collaboration. And now I'll be really happy to answer any question you may have.
Great. So thank you, Wenyi, for your very comprehensive coverage on the PTM, especially ubiquitination, debuquitination, also protein kinase dependent multiplication, etc. Shows the potential to block or modify this PTM to use for cancer treatment. Now we get into the question section, and we let the audience to ask the question first, and the lecture host can ask the question later on. So any question from the audience? So Yi want to take the lead?
Okay, thank you, Wei. That's a wonderful job for your career for so many years, and you have make a huge contribution to the field of post-translational medicine, particularly for ubiquitination issue ubiquitinase. And so my question is that you collaborate with the chemist. Can you hear me?
I can hear you.
Okay, so you collaborate with the chemist and did a lot of work. Wonderful job for PROTAC.
Right.
But the major problem for the PROTAC is all availability because molecular weight is rather big. Okay, and so I know you're a biologist, you're not a chemist, but in the field, it's a limitation for the PROTAC application. All right, and that's why people looking for molecular glue, and which is much smaller, but there's not a good way to identify, the molecular glue. So far all the molecular glue was excellent. So I like to think your idea, whether you think about it, for the future application of PROTAC molecular glue, and how people in the field think about these things. I think many people think about it, but still don't have a good way to have systematically screen for the molecular glue, or how to make PROTAC auto variability. I just like to get your few cents on this issue, which is important for the field, for the application in the cancer therapy.
You are touching the most key question of this field. I agree with you. I believe Albert Einstein said all the best in science are simple and beautiful. I think that's the idea you mentioned. Molecular glue as a small molecule is more druggable, and the structure also look good. But PROTAC look ugly. You have one arm here, another arm here is stretching out. Yeah, it's big, 100%. I think the whole field is now waiting for the 20 PROTAC in clinical trial. If everybody failed one by one, I think actually you might be correct. This is maybe only an academic tool, but it's difficult for translation into really a real meaningful drug. I hold a similar skepticism on this. It's very hard to further reduce the molecular size of the PROTAC. So let's put this way. It's still this, we have to wait. But molecular glue indeed is very exciting. And I heard a really interesting talk from Dan Nomura. So those people, they don't do this. When you do the PROTAC, these both are reversible, right? You have a ligand that reversible binds to it, non-covalent, another ligand non-covalent. But then using one is covalent bond, another is non-covalent. So in this way, you can make this much smaller. And in this way, they can creating, I don't think you can call it a molecular glue, but one hand, they can do covalent bond to the substrate or the E3. In this way, you can make this size much smaller. So I think they are doing that. I feel like this might allow a rational design of future molecular glue. I never thought of that, but I think as long as you keep the one end free, reversible, I think this can be recycled, right? I think one part is covalent bond. There's a new Nature paper show up about 2 weeks ago. They do is that they do this and then they do intramolecule and then make the protein become misfolded. And then decaf 14, decaf 16 will recognize the unfolded protein and then degrading the protein. They don't need to recruit the protein. They may recruit the protein that's too big. I think this is another way to do that. I feel like people are trying to find ways to overcome this. Because if you added a lot of feature onto the PROTAC molecule, it becomes bigger and bigger, and then they become less and less druggable. I think instead of doing the plus, you need to do the minus. There are ways to do that, but I don't know which way better. But I'm also an observer. As you mentioned, I'm pretty much a biologist. I think all the chemical tricks are all initiated by those real medicinal chemists. They are really another way. I don't know whether you noticed. Do biology? We need a 3 year, 4 year to get this done. But they are medicinal chemists. They're talking about 3 months.
So the biology always is behind.
Always behind. Yeah, always behind. They are very fast. Yeah. Okay, but they are all beautiful questions. Yeah.
Okay. I think I'll quit here to give the opportunity to others.
So from the message I received from one audience, he asked you question in Chinese. I just read it for you.
Okay, yeah.
So 根据你设计的 TF Protect 的经验，能否在一个载体上设计多个同样的 DNA motif， 高效靶向的某个滞留性 TF， 比如 MAKE， 或者在一个载体上设计多个不同的 DNA motif， 靶向多个 TF。另外，如何能保证这个 TF Protect 能够高效的进入细胞核内？另外，对于滞留性的 DNA-RNA 结合蛋白，能否也可用 RNA motif 来设计 Protect？
beautiful question. 我觉得他the person is really very, very intelligent. Okay. Number one, we never design more than 1 or 2 or 3. You can do that for CRISPR, but for this, it's difficult. And also the DNA motif are degenerate. So they have certain specificity, but not that specific. So we haven't tried that yet. And indeed, this DNA motif is negative charged and plasma membrane negative charged. So it's harder to get in, but you can using the thing you use for the RNA vaccine, the polymer or something. There's ways to overcome that, but that's beyond my expertise. But in short answer, you can overcome that. You can hijack the recent development in the RNA medicine. Not very hot, right? RNA vaccination, Number 3, he mentioned whether you can do on RNA. Yes, you can. Actually, people began to approach me. They can do it. The only difference is that DNA is relatively stable, and RNA is more unstable. And that can be a reason behind it. We not only can do DNA, we can also do modified DNA. For example, we did recently is methylated DNA. If you use methylated DNA, you can degrading the protein that binding to methylated DNA. You can do M6A RNA. You can degrading all the reader that binds to M6 RNA. So yes, in short answer, you can, but you need to do some chemical trick to block the DNA or the RNA to be degraded, like what the Nobel laureate did. You do introducing one modification, they will not introducing immunity, right? You do something, you can block the RNAs or DNAs to through your decoy, and then that will stabilizing your ligand. Yeah, something like that. We need a case-by-case study. But doable.
So another question he asked or she asked is how can make sure the can be successfully efficiently delivered into the nucleus? The TF
it cannot. Yeah, it is. It's hard. Yeah, I think they need more study. It's hard. Yeah. Because you can see you transfer the first membrane, and then you can transfer the nuclear membrane. As long as you package this in a PEI or liposome, and then they will allow them to deliver. I think some package delivered into the cytoplasm, and some further go into the nucleus. Yeah.
So for the TF you design, can block the MAKE, etc. MAKE is located into the nucleus, right?
Yeah, I actually did not do MAKE degradation. Actually, Craig Cross did it. I reviewed the paper, so he did it. He can degrading MAKE. I was trying to do that. Actually, I failed. I did not get it on. Actually, we tried the CMYC. We tried beta-catenin. We tried NF-κB. We tried E2F1. We were able to degrading E2F1 and NF-κB, but we were not able to get CMYC degraded. But Craig Cruz published the paper. He was able to do it. Yeah.
So in principle, you can block the transcription factor in the cytosol by instead of.
You can. Yeah. If you degrade everything, I mean, true. But some of them can go to the nucleus for sure. Yeah. But this is actually, Jimmy, I mean, long time ago, people have this called the DNA decoy. Actually, we were able to read a lot of papers. So people in the past, they get this DNA motif and they just synthesize a lot and send into the cell. and then this will be decoy. They bind to all the transfer factor. So transfer factor cannot bind into the authentic one. That's called decoy. This has been used for in the past as anti-cancer drug, but we just adding the decoy into another ligand. So turn the passive to active, turn defense into an offense. Yeah, that's what we only did. Yeah.
Great.
But a lot of things need to be done. It's just the first step. Yeah.
So, question from our panel. Yong Chao has any question?
温老师，我有好几个问题想问你一下。第一个问题就是，我想问一下，就是这个泛素化的领域的话，以后的发展方向在什么地方？你在报告里面讲了 E3 的话，有的人讲600多个，有的人讲800多个，但是很多的 E3 的功能都不清楚，但是它的研究的手段也不好做，比如说很多 E3 的，比如抗体啊，各方面都很不好用。所以您觉得这个以后我们这个泛素化领域往哪个方向走？
我觉得现在这个泛素化领域现在的情况就是在往前typical 的 chain linkage， 像 K27、K33、K29 这些typical chain 的那个process 走然后我觉得就是很多的大部分的人现在都在去做 protect 去了我觉得这个 UB-like protein is going to be super exciting 你们在做 methylation 你也还记得吗？我们 actually supposed to be the people that defined the UB-like protein nomenclature for China. I think there's about eight different UB-like protein. 我觉得 more and more there's like ISG, there's ISG, right? There's UFAM, URAM. There's many UB-like protein signaling. I think those signaling nobody does. For example, a lot of those small UB-like modification, they only have one E3. It is relatively easy to study, but their function is understudied, right? I think those are something to think.
第二个问题就是，接着你前面那个问题就是，我们最近也在关注这个不同的这个泛素链，就是发现有一个修饰的话，它是给它加一个杂合的链，就是比如说 K6、K11、K27的这个杂合链。您觉得这个杂合链的话，通过什么来控制它？是通过不同的 E2 来控制，还是某一个 E2 就有这个功能呢
？Actually 这个就是说你可以看到 parking 它 actually 它就可以deliver K6 and multiple chain 那这像我觉得这 100% is the priming. There's a priming E2. I think that's this second E2. I think this is the reason. And also 它的你看这个 linear chain 它只能给 linear， 因为它的这个 composition 就 allow him only do linear. But 我认为是多数是来源于这个 E2。对，这些 really weird 的这个 linkage， 这 weird linkage 的这些 E3 ligase， 像那个 Lubeck parking， 它都是 ring between ring E3 ligase。我觉得 ring between ring allow them to be position in a weird position， 因为你去看那个 K6 是个很 weird 的那个 position， 但是这样 zigzag 的，对，它就是说它必须有两个 ring 的这 hold 这个东西，然后再让它能这个，我觉得这蛮有意思的。还有你要看那个 UB 它那个 Nature paper 可以比 pink one 这个65 phosphorylation sites pink one phosphorylation UB modulating UB 的 function， 我觉得 crosstalk you can think about ubiquitination the UB protein can be phosphorylated, acetylated, sumolated, methylated. All this is like how to say the signaling events. Scaling up the signal events, interplay with the signaling events. 这个就是让这些所有的 signal 就 channel in. Yeah. That's another.
第三个问题就是那个 protype 方面的，就是别看，因为我们现在 protype 的 E3 都用的，比如说是用的是这个康复啊什么之类的。别看这个已经有的这个小分子的话，您觉得哪个 E3 作为这个 protype 一个最有潜力？就是我不管它有没有这个小分子结合的小分子，哪个 E3 您觉得最有潜力呢？
我是这么想的，就整个的 field actually this is a really good question， 用超，我认为这个 field 走错了。 VCHL 是个 tumor suppressor，cereblon 是个 tumor suppressor。So basically， 他们等你把这个 protect 用了以后，2个月以后 ，patient 就会 get rid of VCHL and get rid of cereblon， 因为他 get rid of tumor suppressor 以后 ，number one， 它 resistant to the protect。Number two， it can become more tumorigenic。所以 we got to do is to hijack oncogenic E3， like MDM2， like SKIP2。No， 所以这 tumor cell 是 dilemma。No， 它如果是 get rid of the tumor suppressor， 不是 get rid of MDM2，they die because PV3 go high。如果是不 get rid of it， protect kill them。So either way they are going to die. So I think that's the best way to do. But VCHL although VCHL loss only occurs in kidney cancer, but 如果你去 treat VCHL protect into breast cancer, I'm guarantee you they are going to lose their VCHL in breast cancer, and then become resistant, and then it become more vascular, become like kidney cancer. That's bad. So I think we need to use oncogenic E3. 但是这个 ligand 不好找 ，ligand is more difficult. I think I like your idea. We should search for better ligand.
好，谢谢魏老师。
That's what we are doing now.
Yeah, that's great.
Oncogenic E3, like I said,
that's exactly the idea. Yeah, that should be the best way to go. Yeah, I think. Yeah.
Reading from the meeting chat message, I'm not sure you can read it or not. If you cannot,
let me see. I think I can. Let me look.
There's a question on the top of the list from one person asking you 2 questions. Yeah. Probably can answer number 1 and number 3 questions.
Okay, PDM imposes a wide range of forms. Besides extensive study, isolation, which PM forms do you believe merit further in-depth evaluation in the context of tumor research? Good question. I mean, study methylation. When you say methylation right now, lysine methylation, people study. I think arginine methylation right now very hot. And I really want to see arginine methylation right now really worth of study. And I think parcellation is worth of study because that's PARP inhibitor. I think that's involving DNA damage. That's really important. I love O-glycosylation. Although I didn't study too much yet, but I love it. The reason for that, I think, Jimmy, you know, because O-glycosylation occurs on serine threonine or potentially tyrosine. So basically, the antagonizing phosphorylation events. So you can imagine O-glycosylation derived from glucose, right, and urine, and then this will antagonizing phosphorylation events. So this must be links to cancer and metabolism. So I think that's also a good one to study. I like punctuation because punctuation links to lipid, and as you know, tumor cell loves those different lipid things. So I think that really depends on what kind of cancer types you are studying. I like to study the crosstalk between cancer metabolism and modification. I think all the modification stem from metabolism. Phosphorylation is ATP. Acetylation is acyl-CoA. They all derive from TC cycle. And then you have other modification as well. And I really think we cannot really dissect. I think we need to take everything as a whole. I think I also want to mention protein hydroxylation because I was trained in Bill's lab, and I think protein hydroxylation links to hypoxia. Every tumor experiences hypoxia. I think that's another area worth of study. Sorry, I go too many, but it's really exciting. Yeah,
very, very comprehensive questions. Gopal, do you have any questions?
Oh, number 3, sorry, each type of cells has its own rhythm. Yeah. You mentioned about O-glycosylation. Yes, I love O-glycosylation. I think the O-glycosylation is an interface between glucose level and the nitrogen metabolism, and then kinase signaling pathway. I think that's the most important thing, that the O-glycosylation reader hasn't been found yet. They found the writer eraser, but no reader at all. So imagine this whole system, if you find it, the whole pathway will be revealed ahead of you, in front of you. I think that's actually quite exciting. It's still under study. Yeah.
Good. Hi, Dr. Wei. I'm Gao Beng here.
Oh, hi.
I think very beautiful work regarding the CDK4 inhibitor and the PD-L1 regulation. So I'm wondering, do you have any idea that the PD-L1 regulation is under post-translational modification or in transcription regulation?
Okay, that's a really good question. Let's put it this way. During that short period of time, there are 7 papers published on the same topic. So our work was focused on PD-L1 post-translational modification. But there are papers also showed it can subjecting to transcription regulation, especially interferon pathway. So it can impact epigenetics. So that endogenous retrovirus leads to interferon pathway go high, and that speaks to PD-L1. There's another arm can speak to RB-E12-1, then speak to PD-L1. PD-L1 was almost the center, and is like the center of the universe in immune, and many paths we can speak to it, fit into that. So indeed, it's rather complicated. I think at different time window, there's different modification, but they all, I think, is a cohort of regulation. Let's put it that way. Yep.
Okay, thank you.
Very good. And Dr. Wu Chen.
Hello. Yeah, thank you. It's my turn now. Thank you very much. So, Dr. Wei Wenyi, it's a really very exciting presentation. So I have first a question for following up question for the RNA binding protein degradation. So as you mentioned, we probably can use the RNA for the binding target for the degrader, but due to it's not stable, it's hard to deliver to the cells. So might be not easy, but I was wondering if it is possible that we can use the mRNA expression techniques which we use in the vaccine right now to express the RNA and then degrade the RNA binding protein. So is that possible?
That's possible, but then I think that's a really brilliant idea. You have to figure out another way. I did review a paper that's called in vivo click reaction. So basically, you basically can allow 2 things all being expressed within the cell. Within the cell, this click reaction to put these 2 components together, assemble together. Because PROTAC need 2 parts.
Yeah, that's true. Yeah.
One substrate ligand, one E3 ligand, and then you express your mRNA, but you have to click to that E3 ligand that assemble the whole thing, leading to destruction. So you cannot synthesize the whole thing together. That's not possible. But you can synthesize the mRNA with a special tag, and then you can click it. Reaction potentially can doable. I don't know. You can think about it. There might be some smart way to do that.
Or maybe, I don't know if it's the any kind of E3 ligase or a light tag techniques that has proteins also has the RNA binding or DNA binding characteristics that we can use their their expression levels to express both of these 2 ligands, one for the binding target and one for the E3 or bind, just express. I don't know. It's just the question from
It's really fascinating. I tell you, I was first draw my attention is aptamer. The aptamer nuclear aptamer can. Right now, people are really amazing in developing all kinds of aptamer specific to all kinds of proteins. So they propose to be able to have aptamer for every single protein ever we know. If that's the case, you imagine we can do many things. There's one way you can do turn protein. So you never can turn protein into PCR. But if you can do 1-to-1 ratio aptamer to 1 protein, I can using PCR to detect this. But in the meantime, I can any aptamer to degrade any protein you want. You see what I mean?
This is what I'm doing right now. I'm now
you're amazing. So the idea that how can we really understand protein in a single cell level or little level? The idea is that we can find something that linked to 1-to-1 ratio to the protein, then we can zoom up. If that one is DNA in nature, then we can amplify zillions of folds, and then we'll be able to do that. I think think about aptamer. That can solve a lot of problems. Yeah, using aptamer as a ligand, you can creating all kinds of things you want. Little destruction machine. So the destruction machine is only the entry level. As I mentioned in my last slide, we propose you can do anything you want. If you can drag any enzyme to a protein, you can pretty much fix anything the protein is lacking or you want them to have.
Yeah, thank you very much. But I have one more question.
This is a really good idea. That's a good idea. It's hard for me to think, but it's a good idea. Yeah.
I will try to do that. Thank you. So I have one more question for like the work you have working on the CDK4 regulating the PD-L1 expression. So actually I have read this paper like published on Nature like very carefully before, and it is very beautifully done. So I have a question is that, so besides the regulation of PD-L1 expression, are there any possible mechanism for like how CDK like 4 or 6 inhibitors like you see the palbociclib to enhance the effectiveness of the anti-PD-1 therapy? So besides the PD-L1 expression, like
correct. That's one paper I have to mention is published by Dr. Jane Zhao's group at Dana-Farber. So the idea that once you have CDK4/6 inhibitor, that will lead to reduced DMT1 expression, that leads to reduced demethylation of the endogenous retrovirus elements, that boost the endogenous retrovirus elements expression, that leads to
interferon response, right?
Response. And I know exactly why DMT1 is reduced, because CDK4/6 inhibitor, you will know, they block APC anaphase promoting complex. APC CD12 elevated, that degrading the DMT1. So the all connection is there, all action is there. So indeed, anything you touch epigenetics, you typically involve the endogenous retrovirus, because it's very funny. Our body, 98% of the chromosome are dead fossils of all the previous infection of virus. They are there. They are transposon elements. They are there. They just epigenetic silenced. But if we elevating this process, they will have illusion that the cells like, oh my God, they are invasion. They are virus invasion. Let's elevating the interferon pathway. And then PD-1 will go high, antigen presenting pathway will go high. And this is part of mechanism. Yeah.
Yeah, because we know like the DNMT inhibitor or targeting DNMT like DNA methylation can also like regulate the PD-L1 expression, right? It has been reported. Right?
Not only DMT1, I think LSD1.
Yeah, LSD1.
EZH2. Yeah, a majority, and we tested. We actually have a small library of all the epigenetic regulator, and a big bunch of them can do the same thing. I think this is also kind of, there's a paper published on Nature from a Japan group. They scoop us, by the way. The idea that when you are getting old, you are losing your epigenetic control of yourself, and then your all those things will be elevating, and then PD-L1 is elevated. The elevated PD-L1 will shut down the old people's response to immune surveillance. So that's pretty much explain why we're getting old. We are getting more easy to develop all the disease. Our immune become weaker. That's one my PhD mentor also found. When cells getting cellular level, when cells going to senescence, you have this non-sterile elevation of interferon pathway. That's also epigenetic regulation. So aging, by all means, is an epigenetic phenomenon, because the DNA, nothing changed on DNA level, is the epigenetic regulation. Release my mentor called wakening of the sleeping dog. So the dog was sleeping, and when you're getting 65 years old, they release the power and the heating. I mean, that's what correlates with the biggest risking factor for cancer is whether you are over 65 years old or not. That's the biggest risking factor. And think about that. If that links to epigenetics regulation, PD-L1 expression, damping of your immune surveillance, I think everything began to make sense, right? How can we restore that? I mean, that's another question. How to reverse this process?
Yeah. So and also a lot of work out working on how to degrade the epigenetic writer, eraser, or like a reader, right?
Correct. But one thing I want to really point it out is epigenetics enzymes are really, I mean, we have 600 E3 in our body, so that's the reason we can easily achieve specificity. But epigenetic regulator is very little. So when you're targeting one, you are affecting everything. So you're pretty much ability to kill the tumor, you kill the friends and the foe. You're going to pretty much targeting everything. So we have too few epigenetic regulator in our body. So if you really want to do that, you have to really, really make sure it is tumor specific, not you have to distinguish that. But I think we can distinguish relatively easier for E3 ligase or kinase. Kinase is the same thing. For kinase, we have roughly 600 kinase in our body, and a lot of times the driving events are kinase. I think that's something that we can discuss more, but I think it's just epigenetic regulators too few. So the specificity is getting less, right?
Yeah, thank you very much.
So, Dr. Wei Hong has any question or comments?